So today is the talk that concludes my PhD journey. And I think it couldn't be more perfect than presenting a work that has a story like this, isn't it? When I was telling the story how Dan shaped my decision, I mentioned that one of the main inspirations for the present work was from spatial navigation. So in this video, we see a rat running along an elevated track. Every step the rat takes moves itself from one spot to another, or we can say it transitions from one state to another state, which is illustrated on the right here. At the same time, hippocampal plate cells form unique representations in the spatial axis, telling the rat where it is and how to navigate. From spatial navigation to sensory-motor sequence, path of locomotion becomes a series of manipulations and expressions. And now states are no longer different spots in space, but different configurations of body parts unfolding over time. Well, given the speech or playing a piece is like navigating through a specific path in the sensory motor space. So we know that hippocampal neurons encode states in the axis of space and time. But where are the relevant sensory and motor neurons in the brain? And in what dimensions do these neurons encode movement sequences? Are these dimensions useful for building internal models of physical interactions? Well, the state transitions in the violin playing and human speech seem just so complicated and experimentally intractable. Can we design a task for mice, and which is by the way, convenient model organism in many ways? A task where sensory motor states are just like running on a linear track. Here we are looking at a very thirsty mouse, which is head-fixed, trying to drink water. So this is the bottom view of the mouse, and this is the side view. Here's the mouth, and here's the nose. In front of the mouse is a lick port, but the mouse cannot see it because it's in darkness. The video was captured using infrared camera. A behavioral session consists of many trials. I'm going to show you the high-speed video of one trial played at 20 times the slower speed. Okay, a trial starts with an auditory cue, and the mouse begins to lick. First, at the rightmost position from the mouse's point of view, then the second position on the right, the first position on the right, then the middle position with a straight leg, then the left first position, the left second position, the left third position, which is the end of the sequence. The mouse keeps licking because there will be a delayed water reward coming out. And now you see the droplet, and the mouse continues to lick for a few more times to fully consume the water. So this is one trial. For the next trial, the video on the right, after a random intertrial interval, the mouse starts licking through the same 7 positions, but in reverse. L3, L2, L1, and middle position, R1 is here, R2, R3. Later, we will call sequences like these with adjacent transition port position standard sequence or basic sequences. So you see at the very high level, these are just like an animal navigating back and forth on a linear track and getting rewards on both ends. Well, you may say, Manny, this task is really nothing like violin playing or human speaking, and I don't see how it reflects any human behaviors. Well, I will play another video that will probably change your mind. Right, a disclaimer here. The sequence licking task for mice and the sequence-licking way of playing Rhythm Master, which is a game, were really developed independently. Okay, so much for this. But now imagine the girl levels up in the game, or she wants to learn a variation of the song. Then she will need to know when the change may happen, right? And learn to branch out from the old sequence to handle the new one. In real life, we often deal with a changing environment. Therefore, one has to be able to first detect deviations from what's normal or typical, and change our behaviors accordingly. So can we do it using the mouse's sequence licking task? The answer is yes. So now we are going to change this transition diagram. Take the bottom part as an example. Instead of moving forward from the middle position to R1, R2, R3, and we backtrack the port from the middle position to L2, as indicated by the green arrow. By design, sequences with backtracking transitions like these are less common than standard sequences. So therefore, a mouse won't be able to foresee when the backtracking may happen, and the only way to find out is through tactile feedback. The mouse may say, "Oh, I licked, but the port is not there, and it must be backtracked." Here's a video on the left of an example trial. Now look at L3, L2, L1, and watch here. Backtrack, mouse misses, and relocate the port, and finishes the rest of the sequence. So it turns out mice can learn the backtracking sequences very well. And just to show you how amazingly proficient they are, I'm going to play the same video, but on the right, but with the original speed. So due to some problem with the PowerPoint, I have to exit the presentation mode temporarily, and then play from here. Yeah, that's it. That's in real time. Okay, now I'm going back to the presentation mode. Yeah. Sequence behaviors can have many characteristics. Small chunks of sequences can be found in longer sequences, forming hierarchical structures. Sequences can be flexible, often in response to sensory feedback. Sequences can be planned or controlled based on different behavioral contexts. Many sequences need to be actively terminated, especially for the periodic ones. And most amazingly, sometimes we can generalize a learned sequence to others that have similar structures. Although it seems simple, the sequence licking task in mice features many of these characteristics. So let's go back to the behavior. Here, I'm showing you the behavioral time series of an example trial. It begins with an auditory cue, where the delivery towards the end here, the shaded areas indicate periods of individual contacts between the tongue and the lick port. So we developed the two-axis force sensor to measure instantaneous lick force that the mouse's tongue exerts on the lick port. Here on the top is the vertical component of the force, one for the lateral component. Furthermore, we used deep neural networks to extract the kinematics of the tongue motion. So here the protrusion vector is in red, and the shape of the tongue is covered in pink. From the protrusion vector in red, we can get the length of the tongue L, also here, and its time derivative, or the rate of change, L prime, as well as the angle of the tongue denoted by theta. So as you can see, throughout the sequence, the mouse gradually modulated the tongue angle. In primates, several brain regions have been linked to aspects of sequence behaviors, but Given this new sequence licking task and the little mouse brain, and we wanted to know which brain areas were playing a role in this case. Instead of focusing on a predetermined region, we decided to do a systematic loss of function screen in the dorsal cortex, well, except for the visual part. The most anterior spots include the tongue jaw related premotor area called ALM, as well as part of the tongue jaw subregion of the primary motor cortex, or M1 tongue jaw. M1B is body related M1, which includes limbs and trunk. S1TJ is the tongue jaw subregion of the primary sensory cortex. S1 barofield, or it's just another name for whisker S1. S1TR, S1 trunk, is the trunk S1, which also includes a little bit of TPC. The O regions were manipulated bilaterally. So we used optogenetic stimulation to locally activate channelrhodopsins in inhibitory neurons. By activating inhibitory neurons, we could indirectly silence the nearby pyramidal cells. So later, I will just refer to this process as photoinhibition for simplicity. In order to go around multiple cortical regions in the same mice, we learned a trick to turn the intact skull transparent so that we could point lights anywhere over the dorsal cortex non-invasively and reversibly. Since we're dealing with sequences, it's also interesting to see how inhibition at the different stages of the sequence execution may have different effects. Specifically for inhibition at sequence initiation, light is turned on at the onset of the auditory cue. For inhibition at mid-sequence, the light is triggered when the mouse licks at the middle position, or the fourth touch. For inhibition at the end, the light is turned on at the first consummatory lick after water delivery. Later, we will use the abbreviation init, mid, cons to label the 3 periods respectively. All photoinhibition lasts for 2 seconds. So what I'm going to show you first is what happened when we inhibit S1 tongue jaw region at sequence initiation. So in this plot, x-axis is time. The yellow mark here indicates the auditory cue at the beginning of the trial. This shows the waveform of photoinhibition, and the black traces are the instantaneous lick angle, and the shaded periods are contacts between pores and the tongue as before. So when I play this movie, you will see a vertical line moving along the time axis. That line indicates where the current video frame is. So you see the mouse was able to lick, but licked randomly. Even though the tongue occasionally touched the port, the mouse still couldn't perform and organize the sequence, unless the light is turned off. So we can quantify the consistency in targeting using the standard deviation of angle, or SD angle here, across many trials. Black bars are for trials without photoinhibition, and gray lines, so the cyan bars are those with photoinhibition. Gray lines are individual mice. So you can see that inhibiting S1 tongue jaw caused disorganization in targeting at all 3 periods of time, as shown by the significantly increased variation in lick angle. Surprisingly, despite the disorganized sequences, mice didn't lose the motor capability here to deviate their tongue towards the 2 side, which is quantified using the absolute value of the lick angle with respect to the midline, or abs angle. Mice also didn't lose ability to lick far, which is quantified by the maximum tongue length, L. This reminds me of a classic experiment by Johansen et al., where a volunteer was asked to light a match after her fingertips were chemically numbed. Apparently, her motor capabilities was intact, but the movement became disorganized and uncoordinated due to the loss of sensory feedback. We think that's very similar here, except that we are directly inhibiting the sensory cortical region, presumably cutting proprioceptive information and or touch feedback that are important for generating proper motor command. Now let's turn the light to the tongue jaw motor regions. When the photo inhibition was on, well, we don't see much going on. The mouse was unable to initiate the sequence. And now the light is off, and it could only start the sequence after the inhibition was gone. Trials like this was the majority when we delivered photo inhibition at sequence initiation. But once the sequence was started, it became much harder to shut it down. Here we quantified the same quantities as for S1 tongue jaw, but unlike S1 tongue jaw, photo inhibition in tongue jaw motor region did not significantly change the standard deviation of leg angle. Also, unlike S1 tongue jaw, the inhibition actually impaired the motor capabilities of driving the tongue, as shown by the decreased absolute angle here and the decrease of the tongue length for the first two groups. As a control, photo inhibition in Whiskers 1 did not change any aspects of licking or sequencing. So this is the summary of what we found in this part. So next, we used the multi-channel electrical array to record from many different areas in the different areas of the brain. On the right shows all the recording sites in the dorsal cortex. Given the result from the photo inhibition experiment, we'll be focusing on three areas: ALM, M1 tongue jaw, and S1 tongue jaw. Here in ALM, we recorded 354 single units, and these heat maps are neurons per event time histogram. Each row or each line across the 6 panels is from a single neuron. So these are during right to left sequences, and those are from left to right sequences. So similarly, here shows 235 single unit responses from M1 tongue jaw and 160 neurons from S1 tongue jaw. As you can see that during sequence execution, different groups or clusters of neurons form the sequential firing patterns that tiled the entire sequence. Many clusters show selectivity to leg angle or the direction of ongoing sequence, or a conjunction of both. But to really understand how these neurons are encoding something that's useful for the task, we first need to think about some behavior variables that are important for sequence execution. So the first is the tongue length. So it's an obvious parameter that needs to be controlled. The second is tongue velocity, because it can indirectly control the first order quantity length. And of course, mice lick through different pore positions during the sequence, and that's why they need to change the angle of the tongue. Oh, by the way, these are actually recorded behavioral variables. Red is for left to right sequences, blue is for right to left sequences. Solid lines are standard sequences, and the dashed lines are backtracking sequences, which may not be obvious due to the overlay. Okay, back to this. So directly encoding the tongue angle is not the only way to convey the sequence information. Alternatively, the sequence can encode the following two variables instead of tongue angle. The first is the sequence direction or sequence identity. So throughout left to right sequence, this variable is just a constant, and in right to left sequence is another constant. The second is sequence progress, which is a linear ramp with respect to the goal. So I hope it's intuitive from these three plots that why combining the sequence direction and sequence progress can give us the same information as tongue angle. With the behavioral variables on the left, let's see if we can decode them from the neural activity in different areas of the brain. Decoding analysis involves two steps. First is to fit models that predict each variable using the firing rates of simultaneously reported neurons. So here we just use simple linear regression for that. Once we have these models, these linear models, we can predict behavioral variables at each moment in time, which is what we call decoding. First, let's look at tongue length and velocity. By comparing how similar these decoded traces are with the actually observed ones on the left, as well as the goodness of fit R squared labeled here, so we can see that S1 and M1 tongue jaw are very good at encoding tongue length and velocity, but not so much for ALM. However, when we come to the angle, the first thing we see is the crossing structures. The lick angle goes up in one direction, in one sequence direction, and goes down in the other. But with a closer look, we see a lot of rhythmic fluctuations in the decoded traces from S1 and M1. But the decoded traces from ALM are very smooth, despite similar R-squared values across the three regions. It turns out that M1 and S1 encode an instantaneous version of the tongue angle. You know, the licking goes in bouts, and when the tongue is out, the angle is physically present, and then we can decode it. But in between licks, when the tongue was retracted to the mouth, we can no longer decode that variable. In contrast, ALM encodes the intended lick angle, or an abstract object location. So therefore, the angle information is there regardless of the instantaneous physical state of the tongue. So consistent with the premotor role of ALM, as we move to more abstract variables like sequence directions and sequence progress, we see a better decoding in the premotor and progressively weaker and weaker decoding from M1 and S1. Overall, we can see that S1, M1, and ALM encode behavioral variables with increasing level of abstraction. So from those at the top are closely related to physical states to those at the bottom that are more latent. Although I have to say a good fit in regression analysis shows that the information about a behavioral variable is in the neural activity. However, we don't know if it represents the main dynamics of the entire neuronal population or is only due to one or two very informative neurons, and we were simply cherry picking. There are other methods useful for discovering the main dynamics of population activity in ways that blind to behavioral labels. So one of the most commonly used is a principal component analysis, or PCA. PCA knows nothing about the behavioral variables. It simply looks for the common patterns of activity shared across neurons, which is shown on the right here for this ALM population. And on the left, we just borrowing from previous slide, we combine the decoding of tongue angle, sequence direction, and sequence progress in one 3D plot. So now we can ask, are the decoded trajectories from regression analysis on the left here and the PCA trajectories on the right actually reflect the same neural activities, but they look different only because we are observing them in different reference frames? So this question can be turned into a matching game in 3 dimensions, which is shown here. The algorithm tries to match one set of trajectories with the other by rotating and stretching them. So as a result, the mean canonical correlation coefficient between the pair is 0.95, meaning they are very similar. So here shows what we get when matching the first 3 principal components, but with the decoding of tongue length, velocity, and angle for a S1 tongue jaw population. And this will give us a correlation coefficient of 0.91, which is also pretty high. So the summary plot on the right actually includes all the recording sessions we had for ALM, M1 tundra, and S1. And for each region, we tested different combinations of 3 different combinations of variables with the corresponding principal components. And the take-home message here is that the coding of behavioral variables really dominates cortical activity patterns, and which is consistent with the regression analysis. So far, we have been focusing on neural activity during sequence execution. It turns out that ALM neurons were able to link one sequence to the next over a long intertrial interval. This plot on the top illustrates the pore position during 14 consecutive trials. The yellow vertical lines are the start of each trial. The slopes are sequence executions, and each was followed by an intertrial interval when the pore did not move. Dots represent touches, and notice the scale bar here is 20 seconds. So here shows 2 simultaneously recorded ALM neurons. The red neuron fired persistently only when the pore was on the left. The blue neurons only fired when the pore was on the right. We can decode the pore position using all simultaneously recorded neurons in that session. The prediction in black were very well tracking the pore position in gray. Across all recordings, we have ALM populations differentially fired before the trial start, predicting the target position for the coming sequence. So what's the significance of this? So the significance of this kind of persistent activity is that it links one sequence and with another, thus providing a mechanism to use shorter and more primitive sequences to assemble longer and complex sequences, forming a hierarchical structure. To summarize, we developed a novel sequence leaking task in mice, conducted a systematic loss of function screen to identify brain regions important for sequence generation, and uncovered the coding scheme of this functional cortical network using electrophysiology. So I won't read these, but just want to mention that the points in gray are other interesting findings we had, but I won't be able to present them today due to the limited time. Okay, where do we go from here? I always think imagination is one of the most romantic things our brains can do. But behind this romance is our ability to invoke neural representations of the world even without its presence. And we learned that some representations or states are linked to some other states. Combining both the representations and their links together, we have what's called internal models. So internal models are key for solving complex problems with limited experience. They are also at the very core of creativity, which as scientists makes us who we are and give us the utmost joy in the world, right? Our brain has multiple memory systems. The hippocampal system models locations and episodes in the axis of space and time. Importantly, these models can enable vector calculations which find the path from one place to another, even if those paths were never traveled before. The hippocampal orbital frontal system can represent and link abstract concepts and symbols. And certain parts of the visual system model faces in specific coding axes. In comparison, so we know much less about the coding of internal generative models that link our body parts with the physical world. Since we were born, models of the physical interactions were the very first that we built in our brain. And as we grow up, these models often help us understand more abstract concepts. here like the intuition of an attractor, or the vivid way to think about human relations. So then how can we better study these models of physical interactions? So just like rodents are easier organisms to study hippocampal maps of space and time, they can also be good tools to study the maps of physical interactions. So the new sequence linking task I'm presenting here offers a clear structure of physical states and their transitions. Furthermore, the states include both an internal sensory motor loop inside the mouse, as well as an external moving object, which is the leg port. Although the present work didn't touch on anything about the internal model, I believe what I showed you helped to lay down the foundation for experimentally tackle these questions in mice. Immediately we can ask how representations of individual legs may become embedded in a higher dimensional space after learning. And where does sensory evidence or feedback live in this space? How does it change the course of neural trajectories? Can mice generalize from one sequence to another? And what is the coding space that enables or fails to enable such generalizations in theory? And last, but perhaps the most interesting thing is what circuit structure facilitated these functional organizations. In my wildest dream, I wish this task can withstand the test of time and become a key paradigm for studying the circuit basis of complex sensory motor transformation and internal models of physical interactions. But at the very least, it shows that rodents can have very rich oral-facial behaviors. So they have special ways of exploring the world, though not always the way we are used to think about and anticipate. This is the typical acknowledgement slide I put at the end of the presentation. But I think today is worth going a little